Good morning everyone. Myself Dr. Satish Ramalingam, Associate Professor in the Department of Genetic Engineering, SRMIST Institute of Science and Technology, Katangalattur. Today we are going to talk about cancer gene therapy. So cancer, it's an uncontrolled division of abnormal cells in your body. Cancer therapy includes surgical, radiation, chemotherapy and targeted therapy. So there are emerging therapies like gene therapy, stem cell therapy and immunotherapy. So what is gene therapy? Gene therapy is introduction, removal or modification of a person's genetic code to treat the disease. So here you are seeing the boy in the bubble, David Vetter. So he has a defective ADA gene. Because of that, there is a poor immune system developed in his body. So he has to be remained in the bubble for several years, because when he gets exposed to the air, he gets exposed to the microorganisms, and then that will result in the disease. So he was kept in the bubble for several years actually. And then NASA has helped him to get this suit, astronaut suit. With that he was able to walk outside, but still he is not exposed to the microbes. So this is a condition just because of just one gene, adenosine deaminase gene, that is defective. So he survived for only 12 years because they tried some surgical procedures to transplant his sister's bone marrow into him, but that was not a successful story. However, the science did not stop there. Human genome is made up of 3.2 billion bases, and then there are studies suggesting that diseases happen because of the change in the DNA sequence, and then the interest has come to find a way to edit this DNA so that we can correct several diseases. So there are homology based repair people have identified, and then a Krelox method people have identified to knock down the genes in a tissue specific manner, zinc finger nucleases, talents, and then a CRISPR technology. So these are the several technologies people have developed to edit the DNA so that we can prevent the disease happening. So the next challenge is now we have the corrected gene, how to insert into the cells. So there are several types of vectors available. Scientists have utilized RNA viruses that are called retroviruses, where they remove the disease causing genome and then insert the our gene, correct gene. This has got advantage because there will be integration of the correct gene into the cell efficiently, and there will be long-term gene expression. The disadvantages are sometimes the immunity may affect the gene's expression, and then the specificity is little because the virus can integrate into any cells. So this is the most commonly used one, and then alteration of core proteins are on the way so that we can transduce a specific cell type. So the DNA viruses include adenovirus, adeno-associated virus, herpes simplex virus, and then pox viruses. So here the high transduction efficiency in dividing and as well as non-dividing cells can happen, and then the large packaging capabilities of these viruses, and then we can produce high titers of virus too. So the disadvantage is insertional mutagenesis, because it depends on where this gene of interest is getting integrated into the cell. So that's the only disadvantage. And then this is most investigations are underway to immune specificity of transduction. So there are other technologies like non-viral vectors, like liposomes. So you can encapsulate this gene of interest into the liposomes, and then it can be delivered efficiently into the cell. But some people have tried naked DNA transfections too, and then people have used liposome polycation complexes which can efficiently transfect the cells with the gene of our interest. And people have used peptide delivery systems too. So advantages are large packaging capabilities, and then there is no problem because we are not using any virus here. So disadvantages are low transfection efficiency. So we are not able to transfect all the cells, and then so that limits the expression of genes in the different cells. So the different approaches of gene therapy includes somatic gene therapy and then germline gene therapy. So germline gene therapy is not much actively investigated. However, somatic gene therapy, several people have attempted. So this is further divided into ex vivo gene therapy and in vivo gene therapy. So what is ex vivo gene therapy? So here we take the cells from the patient outside which has the defective gene, and then we already cloned the wild type correct gene. So what we do is that we can transfect using the viral plasmid or something to efficiently transfect the wild type gene into the cells, and then we can selectively grow the population which are having the wild type protein. So these cells are grown more, and then we can inject it back into the patient, so that now their patient's own cell will start expressing the wild type proteins, thereby we can prevent the disease. So earlier I have told about David Vetter, the boy having the problem in the adenosine deaminase gene. Similarly, there are two girls like Cindy and Ashanti. So they both have the same problem caused because of the ADA gene, which is defective. So scientists have done this ex vivo gene therapy where they corrected this ADA defective gene and by using a wild type ADA gene. So after several years, those girls are healthy, and thanks to this technology that scientists have developed. So the next one is in vivo gene therapy. Here, introducing genes directly into tissues or organs without removing the body cells outside. So the challenge is delivery only to intended tissue is a difficulty. Viruses act as vectors for gene delivery, but some inject directly into the tissue. Cystic fibrosis is a disease caused because of a defective gene called CFTR. So this results in the very salty tasting skin. There will be poor appetite, there will be coughing, wheezing, shortness of breath, and then lung infection. These are all caused because of this the defective gene CFTR. So the gene therapy has cured the conditions like this. As you could see in this slide, there are clinical trials where people have given the nebulizer type of gene transfection. So where they use the wild type gene, and then that is efficiently transferred into the lung epithelial cell, and then they started making this wild type CFTR gene that has cured their disease condition. So cancer gene therapy, our objective is to target the cancer cells specifically. So what are the options? To inhibit tumor cell proliferation by inducing restoration of cell cycle control or blocking proteins essential for replications. We can also do to induce cancer cell specific cytotoxicity by introducing suicide genes into the cancer cells. To exploit the property of viruses to selectively replicate and lyse the cancer cells. These are the three commonly used approaches for cancer gene therapy. Among the gene therapies for curing disease, more than 60% of them are for treating cancer. So there are several ways to inhibit the cancer progression. So one is compensation of gene defects. So P53 is a tumor suppressor protein. As the name suggests, this protein suppresses the cancer formation. Unfortunately, more than 50% of the cancer patient has this P53 gene that is defective. So people have used gendicine, which is an adenoviral vector, as I said earlier, and then they have the correct P53 gene, and then when they transfect it into the cell, so the cell started making wild type P53. So this protein suppressed the cancer formation. So next approach is inhibiting the faulty genes in cancer. So the oncogenes are cancer causing genes. So these genes, when they overexpress, they push the cells to divide continuously. So people have used small inhibitory RNA, and then they transfect it into the cancer cells. This goes and binds to the oncogene mRNA and prevents the oncogene protein that is formed. Thereby, the cancer cell progression has been inhibited. So that's one approach. The second approach is like gene therapy based immune modulation. So where they enhance the body's natural immune system to fight against cancer. So Imlygic is a drug where they used a HSV virus, and then they transfected this wild type GM-CSF protein, which results in the immune stimulation. Thereby, the immune system can efficiently inhibit the cancer cell dividing in their body. The next one is suicide gene therapy. Here they introduce gene that makes the cancer cells vulnerable to specific drugs or therapies. So they used a HSV virus, and then they transfect this thymidine kinase protein into the cells. And then when they give a pro-drug, which is not having any effect on all the cells, so these cells, when get phosphorylated because of the thymidine kinase present in the cancer cells, that results in activation of the CAN cyclovir, and then this drug will now kill the cells that are having this prodrug and the thymidine kinase protein. So the clinical trial with the mesothelioma has been successful, where they have cured several patients by this approach. So next one is oncolytic virotherapy, where they use viruses that selectively kill the cancer cells. So Onyx 015, it's adenoviral based, and then there is a protein called E1B. So when the scientists mutated this protein, the cancer cells, usually I told you there is 50% of the cancer cells that don't have the 50% of the cancer patients don't have the wild type p53 protein. So in those patients, when the p53 protein is not wild type, it's mutated, so it cannot suppress the cells dividing. So the virus replicates and kills these cancer cells that are transfected with the E1B mutation, and then because there is no p53 wild type protein to inhibit the cell cycle, so these cells will be killed selectively. So these are oncolytic viral therapy. RBM3 is a proto-oncogene. The overexpression of this protein increases cancer cells to divide. So if you could see, the siRNA based, small inhibitory RNA based approach has prevented the tumors growing, whereas the control cell cancer cells, they have been growing in animals, and then they form huge tumor. So knockdown of the gene has been shown promise here. This figure shows the suicide gene therapy. You have HSV viruses which has the thymidine kinase protein. When it is transfected into the cell, so they go in, and then if the cells are treated with the prodrug ganciclovir, so if the cells has the thymidine kinase and they have this prodrug, so it acts on it and then phosphorylates this ganciclovir drug and then makes the active drug. Once the drug is active, it will kill the cells expressing this thymidine kinase. So in this way, we can efficiently kill the cancer cells alone if we try to express this thymidine kinase HSV plasmid into the cancer cells. So this figure demonstrates how oncolytic virus can be used to kill the cancer cell. So normal cells, when the virus infects, there is no replication and damage the cell. But in the tumor cells, when there is a virus infected, the virus replicates and then it kills only the cancer cells. Thank you for your attention. I hope you have understood about the gene therapy and then how it can be used to treat cancer. Thank you.